Verotoxin is one of the most potent bacterial toxins produced by enterohemorrhagic Escherichia coli, also known as EHEC. The other name for it is Shiga-like toxin because of its close relation to the Shiga toxin. Despite the happy-looking Patrick star, it is not a very happy and pleasant disease. Patrick will come up later in the video. Verotoxin consists of 5 identical B subunits and 1 A subunit. Part of the A subunit sits in the center of the B subunits. The A subunit splits into 2 fragments, A1 and A2, once it reaches the inside of the cell, each having 2 different functions. Taking a closer look at the A subunit, A1 and A2 are connected by a disulfide bond between the 2 cysteine residues. Once this bond is broken, the A subunit separates into 2 pieces, A1 and A2. This breakage enzymatically activates the A1 subunit. Its function is to enzymatically remove one adenine base from the structure of ribosomal RNA, inhibiting its function to elongate peptides. This stops protein synthesis completely. The function of the A2 subunit is to attach the A subunit to the B subunit. Now let's take a closer look at the B subunit. If we look at this toxin from a different angle, we can see that the B subunit itself resembles a star. The B subunit is responsible for the binding of the toxin onto the cell membrane. This is where Patrick comes in. He's very close to the B subunit shape. Each of the points in the star are an identical B subunit, making a total of 5, known as a pentamer. Each of these subunits contains 2 sets of 3 antiparallel beta sheets linked together on both sides, making a beta barrel, as well as an alpha helix located between beta strands 4 and 5 of each monomer. The B subunit binds to a glycosphingolipid receptor, GB3, on the outer membrane of a cell. This receptor has 3 globotriose binding sites per B subunit monomer, making a total of 15. These sites are identified as Site 1, 2, and 3 on each B subunit. Site 1 is the cleft found between the phenyl groups belonging to phenylalanine on one monomer and an aspartic acid on the adjacent monomer. This site is fully exposed to the solvent because it is lying on the outer edge of the B subunit. Hydrophobic interactions are observed here. Site 2 is defined by an asparagine, an arginine, and a glycine, all on the same monomer, which are all hydrogen bonded together. The asparagine sits on the N-terminal side of the alpha helix, the arginine connects the alpha helix and the beta sheets together, and the glycine is where the beta sheets of the first antiparallel strand end. Finally, Site 3 involves just a tryptophan, which sits close to the N-terminal of the alpha helix, pointing away from the center pore of the B subunit monomer. This allows the hydrophobic indole rings to interact favorably with each other. There aren't quite as many hydrogen bonds here because this interaction is more dependent on the hydrophobic interactions. Within humans, verotoxin specifically targets the gastrointestinal epithelial cells found in the lining of the intestines. These are the cells that have the GB3 receptor on them. GB3 is a glycosphingolipid receptor made up of alpha-galactose 1,4 linked to beta-galactose linked to 1,4-beta-glucose. Cells that express more GB3 receptors on their surface feel the effects more potently because the toxicity is highly correlated with binding affinity. As previously mentioned, the B pentamer is going to be the one that binds to this receptor. Once verotoxin has attached the GB3 receptor, the A subunit enters into the cell through endocytosis. Once inside the cell, the vesicle is invaded by lysozymes. The lysozymes are able to cleave the disulfide bond holding together the 2 parts of the A subunit, and the A1 subunit becomes activated. It is now able to bind to ribosomes by removing an adenine base from its structure, preventing it from being able to elongate peptides. Removing the adenine base inactivates elongation factor 2 by ADP ribosylation, which is the addition of ADP ribose to a protein. This is an irreversible post-translational modification and inhibits peptide chain elongation. This leads to cell death because the cell can no longer manufacture proteins. As you can imagine, this is a major problem within your digestive tract and can lead to very serious problems such as hemolytic uremic syndrome, or HUS for short. This toxin also heavily impacts the renal system as well as vascular cells. It does this by moving into the bloodstream with the aid of a molecule called intimin. Intimin promotes the efficient transport of verotoxin from the intestines into the bloodstream. Being infected by verotoxin is often the result of eating contaminated hamburger meat and initially causes diarrhea. In about 10% of the cases, however, it leads to more serious consequences, such as hemolytic uremic syndrome. Hemolytic uremic syndrome is characterized by a common triad which is commonly shown to result in patients: hemolytic anemia, which is the rupture of red blood cells, thrombocytopenia, which is another term for low platelet count, and acute renal failure, which is when the kidneys stop functioning. So how does one with this disease get help? Antibiotics was the first attempt at solving this problem. However, it led to even more toxins being produced, which is not a very efficient way to combat this disease. Taking antibiotics also increased the patient's risk for developing more serious complications, such as hemolytic uremic syndrome. Synsorb PK was the next attempt. It is classified as a diatomaceous silicone diamic compound, meaning that it was made of naturally occurring silicone found in the earth. It was shown to avidly bind and neutralize the toxin, however, it was not able to reduce dialysis or help fix renal complications in general. Starfish was the next new and improved Synsorb PK, which bound 1,000 times better to the toxin. In testing, it was shown to protect mice against a lethal dose of Verotoxin, however, the window of time it had to be administered was too specific. Sorry, Patrick. Finally, antibiotics that were A subunit specific were developed against Verotoxin. This was shown as the best option because in testing it protected kids against hemolytic uremic syndrome development, the best option for preventing. It is still in the development process, but the future of treatment seems promising. Stay tuned.